Okay, let's unpack this. Imagine a towering redwood tree scraping the sky, or even just, the crisp snap of a celery stalk. Now picture something like a jellyfish.
Right. Completely different forms.
Exactly. The fundamental difference, what gives plants their incredible rigidity, their shape, and really their ability to reach for the sun, it comes down to something you might think of as simple, but is, well, anything but the plant cell wall.
It really is the unsung hero of the plant world. It's this dynamic, incredibly complex structure that enables pretty much everything we associate with plant life.
It really does. And that's our mission today. We're taking a deep dive into the absolutely fascinating world of plant cell walls. We're drawing our insights from a key chapter in a major textbook, Plant Physiology and Development.
Foundational text.
Definitely. We've pulled out the most important knowledge nuggets from this source material, and we're going to try and unpack everything about what these walls are made of, how they're built.
How they manage to grow.
Yes. How this seemingly rigid barrier allows plants to grow so dramatically and, why it matters not just to plants, but actually to us.
Oh, absolutely. You'll discover how the wall provides strength, controls shape, acts as a defense system, and even links plants directly to, well, vital global processes like carbon storage and the materials we use every single day.
Okay, so let's jump right in. What is a plant cell wall? It's clearly more than just a rigid box, right?
Oh, much more. You could think of it as a really sophisticated composite material, maybe like rebar and concrete, but biological and crucially highly dynamic.
Okay.
It sits outside the plasma membrane, the cell's outer boundary, and it's built primarily from the scaffold of strong rope-like cellulose fibers. These are embedded within a sort of jelly-like matrix of other complex sugars, polysaccharides, plus proteins, and other polymers too.
And this is the really big difference compared to animal cells, isn't it? We don't have this external layer.
Precisely. And because we don't, our cells are generally flexible. They can move around. Plant cells, on the other hand, are fixed in place, and they get their structure almost entirely from this wall. It's a fundamental difference that shapes everything from how plants develop tissues to how individual cells actually function.
So besides just providing structure, what are the wall's main jobs? It sounds like it does a lot.
It does so much. I mean, first, it protects the delicate cell contents inside the protoplast. It determines the cell's shape, and it prevents the cell from bursting when water rushes in due to osmosis. That's a key function shared with walls in bacteria and fungi, too.
Right. Keeps it from exploding.
Exactly.
Yeah.
But uniquely for plants, the wall provides that incredible mechanical strength needed to, well, stand tall against gravity. Think of the trunk of a massive tree that's all thanks to cell walls.
Wow.
It's also the glue, effectively, that holds neighboring cells together. This is absolutely essential for building organized tissues and organs. Without cell adhesion, plants would just be loose collections of cells.
So it's literally the scaffolding and the glue.
Pretty much, yeah. It also plays a critical role in controlling cell shape and size during growth, what biologists call morphogenesis. The wall's ability to expand in very specific, controlled ways is fundamental to how a plant develops its form.
Okay, so it's not just static structure. It's involved in growth control.
Definitely. And it works hand in hand with the high internal water pressure inside plant cells that's called turgor pressure. The wall acts like a strong container, like a cellular exoskeleton, allowing this pressure to build up.
Ah, like a tire needing both the rubber and the air inside to hold its shape and function.
That's a really good analogy. The wall is the robust outer layer, the tire, and the turgor is the air pressure that makes the whole system rigid. This rigidity is vital for everything holding leaves up to catch sunlight, keeping the plant upright, and even helping drive water transport.
Okay, that makes sense.
And speaking of water transport, the wall reinforces the plant's plumbing system, the xylem vessels. It prevents them from collapsing under the strong suction, the negative pressure, that pulls water upwards from the roots.
So it keeps the pipes from caving in.
Exactly. It also acts as a selective filter, controlling which molecules can pass through to reach the plasma membrane based on size and even electrical charge. And it can even sense the cell's integrity. There are proteins linking the wall to the membrane that signal if the wall is stressed or damaged.
Wow. Okay, it's like a structural engineer, a plumber, a filter, and a security guard all rolled into one.
And it's definitely key to defense against pathogens, parasites, even herbivores. Sometimes fragments of the wall that get broken down during an attack can act as alarm signals, triggering wider defense responses throughout the plant.
That's amazing signaling. And you mentioned its importance to us directly.
Oh, hugely important. Every piece of paper you use, every cotton shirt you wear, linen, all lumber and wood products, these are fundamentally cell wall materials. primarily cellulose.
Right, the raw materials.
Exactly. And there's a massive effort now in converting plant biomass, which is mostly cell wall material, into biofuels. The cellulosic ethanol. It's also the most abundant source of organic carbon on the planet. A major way plants store the carbon they capture from the atmosphere through photosynthesis. So it's absolutely crucial in global carbon cycles.
It's incredible how one structure is so vital biologically and economically. If you look at it under a microscope, though, you see that walls aren't all the same, are they?
That's right. Their structure varies a lot depending on the cell type and its specific function. Cells that are mainly for storage, like in a potato tuber, might have relatively thin walls. Yeah. But cells that need a lot of strength, like the fibers in flax or the cells in Wood's parenchyma or xylem cells, they have much thicker, heavily reinforced specialized walls.
And even within one cell.
Yes. Even different sides of the same single cell can have walls with different structures or thicknesses. Like an epidermal cell on a leaf. The outer wall might be thicker, maybe coated with a waterproof cuticle, while the inner walls connecting to other cells are different. This reflects the cell's specific job and its polarity, its directionality.
And there are two main types of walls, generally speaking, based on when they form during the cell's life.
Yes, that's a key distinction. The primary wall and the secondary wall. The primary wall is the first one formed by cells while they are actively growing and expanding. So it needs to be strong, but also flexible and extensible. It's usually thinner, though not always.
Okay. Formed during growth.
Yeah.
And the secondary wall.
That gets deposited after the cell has stopped growing, after it's finished expanding. It's laid down inside the primary wall, between the primary wall and the plasma membrane. It's often much thicker, much more rigid, and provides permanent strength. Secondary walls are really characteristic of cells whose main job is structural support or water transport, like those found in wood tissue tracheids, vessels, fibers. And these secondary walls are frequently reinforced with another complex substance we need to talk about, lignin, though not all secondary walls are lignified.
Right, lignin that makes wood woody. So primary walls for growth and flexibility, secondary walls for strength after growth stops. And you mentioned something between the cells, too, the glue.
Yes, that's the middle lamella. It's a very thin layer, kind of sandwiched between the primary walls of adjacent neighboring cells. It's particularly rich in sticky polysaccharides called pectins, and maybe some specific proteins, too. It originates from the cell plate during cell division and acts like a flexible cement, basically holding the cells together.
Got it. Okay, let's dig into the building blocks then. What are these walls actually made of at the molecular level?
Okay, the main components are polysaccharides, which are just long chains of sugar molecules linked together, and also some structural proteins.
Polysaccharides, right.
The absolute star of the show is cellulose. It forms these incredibly strong, cable-like structures called microfibrils, which are the main reinforcing elements.
A rebar, basically.
Kind of, yeah. Then you have the matrix polysaccharides. These fill the space between the cellulose microfibrils. The two main types are pectins and hemicelluloses.
Pectins and hemicelluloses. Oh.
Pectins are really complex, diverse, and they love water. They're hydrophilic. They form gels and are particularly abundant in primary walls, acting like a hydrated filler. Hemicelluloses are also a diverse group of sugar polymers. They tend to bind quite tightly to the cellulose microfibrils and help cross-link them, creating a coherent network.
And proteins.
Yes, there are various structural proteins embedded in the wall too, especially in primary walls, maybe 2 to 10% by dry mass. Their exact functions aren't always clear, but they likely play roles in strengthening the wall or modifying its properties. And there are some very specialized ones like AGPs, arabinogalactin proteins, which might be involved in signaling.
And you mentioned earlier that tiny details, like how the individual sugar units are linked together, make a huge difference in the properties of these polysaccharides.
A dramatic difference. It's
Yeah.
incredible. Take glucose, the simple sugar. If you link glucose units together with one type of bond, an alpha linkage, you get amylose, which is a component of starch, soft, digestible, used for energy storage.
Right, like in potatoes or rice.
Exactly. But link the same glucose units with a different bond, a beta linkage, and you get cellulose, incredibly strong, rigid, indigestible for us, perfect for building structures. Link them with yet another bond, beta-1,3, and you get callus, which is involved in wound responses. So just changing that one chemical connection completely alters the molecule's shape and function.
That's fascinating, just the tiny tweak in the chemistry.
It really is, and it applies to all the different wall polysaccharides. The specific linkages determine their shape, how they pack together, how they interact with water and other polymers, and ultimately their role in the wall.
Okay, so how do these components actually get assembled into a functional wall? Where does this happen?
Right, this is where it gets really dynamic. The synthesis is quite compartmentalized. The cellulose microfibrils, the main structural cables, are synthesized right at the cell surface, at the plasma membrane.
Not the membrane itself.
Yes, there are these large protein complexes embedded in the plasma membrane called cellulose synthase complexes, often visualized as rosettes. They grab sugar building blocks, specifically UDP glucose, from inside the cell and essentially spin out the long cellulose chains directly into the wall space, where they immediately associate with other chains to form those strong microfibrils.
Wow. So the wall is literally being built from the inside out, right at the very edge of the cell.
Exactly, for the cellulose component. It's quite amazing to visualize these enzyme machines moving in the membrane, laying down tracks of cellulose.
And the other stuff, pectins and hemicelluloses in the matrix.
They're made differently. They're synthesized inside the cell, primarily within the Golgi apparatus part of the cell's internal membrane system.
Okay, so made deeper inside.
Right. Inside the Golgi, various enzymes called glycosyltransferases assemble these complex pectin and hemicellulose polymers. Once made, they get packaged into little membrane-bound sacs called vesicles.
Cool delivery packages.
Exactly. These vesicles then travel to the plasma membrane, fuse with it, and essentially dump their contents, the pectins and hemicelluloses, out into the wall space outside the cell. This process is called exocytosis.
So it's a really coordinated effort, cellulose being spun out at the membrane, while the matrix components are made inside and shipped out via these vesicles.
Precisely. It's a continuous process, especially in growing cells. Synthesis, delivery, and then assembly into the wall structure are all happening concurrently.
So once all these components are outside the cell, in the wall space, how do they actually assemble into a functional, strong network? They don't just float around, right?
No, definitely not. They need to achieve a specific physical arrangement and form bonds, both non-covalent and sometimes covalent, to give the wall its strength and yet allow for controlled expansion. The exact details are still being worked out, but several factors are involved.
Like what?
Well, there's some degree of self-assembly. Hemicelluloses, for instance, naturally tend to stick to cellulose surfaces. Pectins can form gels on their own, especially if calcium ions are present.
So they just sort of naturally find their places?
To some extent, yes. But the interactions seen in vitro, just mixing components in a test tube, often aren't as strong or specific as what seems to happen in vivo in the actual wall. So enzymes are almost certainly involved in properly integrating and potentially modifying these polymers once they're secreted.
What kind of enzymes?
There's a really interesting class of enzymes called xyloglucan endotransglucosylases, or XETs. Xyloglucan is a major hemicellulose in many plants. XET enzymes can actually cut a xyloglucan chain and then attach the cut end to another xyloglucan chain.
So they cut and paste these chains.
Essentially, yes. This could be a way to integrate newly secreted xyloglucans into the existing wall network, potentially creating new connections or modifying existing ones. It might help strengthen the wall or allow for rearrangements during growth. There are likely other similar enzymes acting on other polymers too.
And you mentioned calcium and pectins earlier.
Right. Pectins, especially a type called homogalacturonin, have acidic groups. Enzymes in the wall called pectin methyl esterases, PMEs, can remove methyl groups from these pectins, exposing negative charges.
Okay.
And if calcium ions, which are positively charged, CET2+, are present, they can form bridges between two negatively charged pectin chains. This creates a sort of egg box structure, cross-linking the pectins into a much stiffer gel. This calcium cross-linking is thought to be really important for cell adhesion in the middle lamella and for making the primary wall less extensible, especially when growth stops.
So controlling the pectin chemistry affects the wall stiffness.
Absolutely. And there are other potential cross-linking mechanisms too, involving phenolic compounds or proteins, often catalyzed by enzymes like peroxidases. It's a complex interplay of deposition, self-assembly, and enzymatic modification.
Okay, let's switch gears slightly to cell expansion. How does this wall, which provides rigidity, actually allow the cell to grow, sometimes massively?
That's one of the most fascinating aspects. Primary walls have to be strong enough to withstand the cell's internal turgor pressure, but also extensible enough to allow the cell to enlarge, often by a huge amount, 10, 100, even 1,000 times or more in volume.
Wow, how does it not just break?
Well, it requires a constant coordinated process. New wall material is continuously synthesized and secreted into the wall, integrating into the existing structure, while the wall simultaneously stretches and yields to the turgor pressure. The key is that the wall undergoes controlled loosening.
Loosening.
Yes. The wall doesn't just stretch elastically like a rubber band. It undergoes irreversible yielding, or creep. There have to be mechanisms that allow the load-bearing connections within the wall to shift or break and reform, allowing the network to expand.
And that's where those expansin proteins come in.
Exactly. Expansins are major players in this wall loosening. We know that growing plant tissues extend much faster if you place them in acidic conditions. This is called acid growth.
Acid makes them grow faster.
Yes, and it turns out this is because expansin activity is optimal in acidic pH. These proteins are found in the cell wall, and they seem to work by disrupting the non-covalent bonds, the stickiness, between cellulose microfibrils and the matrix polysaccharides, particularly hemicelluloses like xyloglucan that coat or tether the microfibrils.
So they're not cutting the main chains, just sort of unsticking them.
That seems to be the primary mechanism, yes. They don't break down the polymers themselves, but they allow the existing network components to slide past each other a bit under the force of turgor pressure. This allows the wall to stretch irreversibly.
So they act like molecular WD-40, loosening things up.
That's a pretty good analogy. They reduce the stress in the wall by allowing it to yield. This yielding is crucial because it lowers the cell's turgor pressure slightly, which in turn lowers the cell's water potential.
So the cell can then take up more water.
Precisely. Water flows in, the cell volume increases, stretching the loosened wall. It's a cycle. Wall loosening leads to stress relaxation, which drives water uptake, which causes expansion, stretching the wall components until they become taut again, ready for another round of loosening. Without this stress relaxation, the cell would just build up pressure or thicken its wall, but it wouldn't actually expand in area or volume.
That makes sense. It's the loosening that enables the expansion. Okay, so expansins help control the rate of growth by allowing loosening. But how does a cell control the direction of growth? Why do some cells get longer and skinny while others grow more like a sphere?
Great question. While the turgor pressure pushing outwards is pretty much equal in all directions, the wall itself is usually not equally strong in all directions. Its mechanical properties are anisotropic.
Anisotropic, meaning different depending on the direction.
Exactly. And the primary determinant of this anisotropy is the orientation of the cellulose microfibrils. Remember those strong, cable-like structures?
Yeah.
They provide the main resistance to stretching.
Right, the rebar.
So if the microfibrils are deposited in a more or less random, tangled orientation, the wall is roughly equally strong in all directions, and the cell will tend to expand isotropically like a balloon, becoming spherical.
Okay.
But if the cell aligns the deposition of new cellulose microfibrils predominantly in one direction, say, wrapping them around the cell like hoops around a barrel, then the wall becomes much stronger, much more resistant to stretching in that hoop direction.
So it can't easily get fatter.
Right. It resists expansion and girth. But it can still stretch more easily in the direction perpendicular to those hoops along the length of the barrel. So this transverse alignment of microfibrils restricts radial expansion and promotes elongation. This is fundamental to how plant stems and roots get longer.
So the pattern of the cellulose rebar dictates the shape change. How does the cell control where those microfibrils get laid down?
That's where the cell's internal scaffolding, the cytoskeleton, comes into play specifically. Microtubules. These are tiny protein tubes located just inside the plasma membrane, in the cytoplasm.
Microtubules.
Numerous studies have shown a remarkable correlation. The orientation of the cortical microtubules, those near the cell cortex or edge, often mirrors the orientation of the newly deposited cellulose microfibrils just outside the plasma membrane.
So they line up.
They often do, yes. If you look at an elongating cell, you'll frequently see the microtubules arranged in hoops around the cell, parallel to the hoops of newly made cellulose.
And if you mess up the microtubules.
Great point. If you treat cells with drugs that disrupt microtubules, or look at mutants with defective microtubule organization, you often see disorganized cellulose deposition. The microfibrils are laid down randomly.
And the cell shape changes.
Yes. Instead of elongating properly, the cells tend to swell up isotropically, becoming more spherical or bulbous. This is strong evidence that the microtubules somehow guide the deposition of cellulose.
How do they guide it? Are they like train tracks?
That's the leading hypothesis. The idea is that the large cellulose synthase complexes, those rosette machines we talk about that make cellulose at the membrane, are somehow guided along the paths laid down by the cortical microtubules. Perhaps they are physically linked.
Wow.
There's even evidence suggesting that the cellulose synthase complexes are delivered to the plasma membrane in vesicles that initially associate with microtubules. And proteins have been identified, like one called CSI1, that seem to physically connect the synthase complexes to the microtubules.
That's an incredible level of molecular coordination. The internal cytoskeleton is dictating the pattern of the external wall construction, which then determines the direction of cell growth.
It's a beautiful example of how different cellular components work together to achieve a complex outcome, like directed cell expansion. It involves signaling, cytoskeletal dynamics, enzyme activity, polymer synthesis, and biophysics, all orchestrated.
Okay, we've covered primary wall growth. What happens when the cell stops expanding? You mentioned the wall rigidifies, and then the secondary wall might be laid down.
Right. Once a cell reaches its final size and shape, the whole process of primary wall expansion needs to shut down. Growth cessation involves several changes that make the wall less extensible.
How does it get stiffer?
Well, the activity of wall loosening factors like expansins likely decreases, but also cross-linking within the wall can increase. We talked about calcium bridging and pectins that can definitely make the wall more rigid. And in some cell types, especially grasses, specific types of polysaccharides might be removed or modified. Plus, enzymatic cross-linking of phenolic compounds or proteins via peroxidases can occur, essentially locking the structure in place. Sometimes multiple mechanisms contribute to this final rigidification.
And then the secondary wall gets deposited.
Often, yes,
Yeah.
particularly in cells specialized for support or water transport.
Yeah.
As we said, it's laid down inside the primary wall. It's typically much thicker, and its composition is different,
Different how?
usually much higher in cellulose content. And the hemicelluloses are different too often, types like vilans or glucomannans that have fewer side branches, allowing them to pack very tightly with the cellulose microfibrils. This dense packing contributes significantly to the strength and rigidity of the secondary wall. And of course, there's often lignin.
Right, lignin. Tell us more about that. It sounds crucial for secondary walls.
It absolutely is for many types. Lignin is a very complex, irregular polymer made from phenolic building blocks called monolineals. These are synthesized inside the cell and then transported out into the wall.
Okay.
Once in the wall, enzymes like peroxidases and lactases oxidize these monolineals, creating reactive radicals that then randomly link together, forming a large, branched, hydrophobic polymer network.
Randomly linked.
Pretty much. Unlike the precise structures of cellulose or proteins, lignin doesn't have a defined repeating sequence. It fills the spaces between the cellulose and hemicellulose, kind of like embedding the polysaccharide framework in a hard plastic-like resin.
And that makes it strong and waterproof.
Exactly. Lignification drastically increases the wall's compressive strength and stiffness. It also makes the wall hydrophobic. It repels water, which is essential for efficient water transport through xylem vessels, preventing water from leaking out. And it makes the wall highly resistant to degradation by microbes and enzymes, which is why wood is so durable.
So it transforms the wall from that potentially flexible primary state to something really robust and resistant.
Precisely. It's a process of infiltration and reinforcement. Lignin deposition often starts in the middle lamella and primary wall at the cell corners, and then spreads throughout the secondary wall layers.
Layers. Secondary walls have layers.
Yes, often they do. Secondary walls are frequently deposited in distinct layers, typically called S1, S2, S3, with the S2 layer often being the thickest. And interestingly, the cellulose microfibrils within each layer usually have a consistent but different orientation compared to the adjacent layers.
Like plywood.
That's a fantastic analogy. The alternating orientation of the cellulose grain in different layers provides strength in multiple directions, making the overall structure incredibly strong and resistant to splitting. The S2 layer, often with microfibrils aligned nearly parallel to the cell axis, is particularly important for tensile strength.
Wow. Okay, this has been a really deep dive. We've gone from the basic definition and functions, structure, shape, protection, adhesion, growth control, filtering, defense.
And its huge economic importance.
Right. To the key components, cellulose, pectins, hemicelluloses, proteins, lignin, and then how they're made cellulose at the membrane, matrix stuff in the Golgi.
And how they're assembled and modified by enzymes like XCTs and PMEs.
Then we got into the mechanics of growth, the role of turgor, the crucial wall loosening by expansins driven by acid pH.
And how that stress relaxation allows water uptake and expansion.
And how the direction of that growth is controlled by the orientation of cellulose microfibrils, which in turn is guided by the cortical microtubules inside the cell. That coordination is just amazing.
It really is.
And finally, the transition to secondary walls after growth stops, with their distinct composition, layered structure, and often lignification for ultimate strength and durability.
We've really covered the key physiological processes, the developmental timing, the molecular mechanisms involving genes like CISA and enzymes like expansins, drawn on experimental evidence and models, and link it all back to the real-world functions and applications of these walls. It really emphasizes that the plant cell wall isn't just some static box. It's an incredibly dynamic, complex, and essential biological material.
Absolutely. It's fundamental to plant life as we know it, enabling everything from microscopic shape changes to the existence of the tallest trees on Earth. We've definitely done a comprehensive summary of the key points from that source chapter.
Yeah, I think we've hit all the major concepts.
So, thinking about all this complexity and adaptability, how plants fine-tune wall composition and structure for different cells, different stages, different stresses, it really makes you wonder, doesn't it? What other incredible, maybe hidden ways might these cell walls be working to help plants survive and thrive, especially in a changing world?
That's a great question. Or, flipping it around, considering how even slight changes in those molecular linkages create vastly different properties. Imagine the possibilities if we could truly understand and engineer these microscopic structures with precision. What new materials could we design, or how could we enhance plant resilience? There's still so much to explore.